The Origin and Evolution of Repetitive DNA Sequences. My doctoral thesis. As a doctoral student at UC Irvine, UCI, I was privileged to play a significant role in the discovery of a fundamental mechanism by which repetitive sequences arise and expand genomic DNA, which I called slipped-strand mispairing, SSM. Often called replication slippage, the original SSM publication is now widely cited by the scientific community and well supported by empirical evidence. As is so often the case with scientific discoveries, serendipity and trial and error, including pursuit of false leads, played important roles in my discoveries. Throughout my undergraduate studies at UC Berkeley, UCB, and extending into my graduate research at UC Irvine, I was intrigued by the role that development plays in the evolution and reproduction of complex organized structures and functions. I began working with fruit flies, the powerful model system that ushered in the revolution in evolutionary developmental biology. I was working with a DNA sequence found in fruit flies that exhibited cross-hybridization with sequences in other species that had been implicated in determining gender, sex determination during development. Since cross-hybridization is often due to evolutionary conservation of homology, evolutionary descent, I had hoped to learn more about the genetics of sex determination in fruit flies. As fate would have it, this was a thoroughly false lead. Not only did this fruit fly sequence have nothing to do with sex determination, it was also cross-hybridizing not because of homology, but rather because it contained simple repetitive DNA sequences. These simple sequences included a variety of long tracks of short tandem repeats, such as GATA, GATA, GATA. Although the accidental discovery of this artifact felt like a disaster at the time, the good news is that I became fascinated with the evolutionary origins of simple repetitive sequences, which were starting to turn up in a broad range of organisms, particularly in non-protein coding DNA, that is, sequences that do not specify amino acid sequences of protein chains. Against the strong objections of my principal investigator, I decided to change the direction of my doctoral thesis, and to do so, I needed to find a new mentor. Fortunately, I found George Gutman, who kindly provided both resources and guidance for a thesis that fell outside of the main focus of his own laboratory. I was astounded to find that not only were simple repetitive sequences extremely common in non-protein coding regions of virtually every eukaryotic species that I looked at in the database, including a broad range of plants and animals, as well as humans, but surprisingly, no one had published a good explanation for how they arise in the first place. Presumably, these repetitive sequences were produced by enzymes that synthesize DNA. But how? One possibility was that they were produced by the enzymes that routinely replicate DNA, known as DNA polymerases. I hypothesized that short, simple runs of bases that initially occur randomly, for example a run of GGGG or AAAAA, would be prone to polymerase errors, in which the two strands of replicating DNA are accidentally mispaired during replication, which can result in the insertion or deletion of repeat units. Not only that, but the longer the repeat, the greater the chances of mispairing. Therefore, production of simple repeats by slipped-strand mispairing, also known as replication slippage, might be a self-accelerating process, under conditions that favor, or select for, insertions more than deletions. I went about testing several of these assumptions by observing the expansion or contraction of synthetic repetitive sequences that I introduced into clones, that I introduced into bacteria, using recombinant DNA technology. This provided a model system where I could systematically detect and subsequently sequence newly occurring insertions or deletions. I found that not only did these simple cloned sequences expand, but they expanded at extraordinarily high rates. These early studies laid the groundwork for numerous subsequent studies by other scientists. Synergies between replication slippage and point mutations. Point mutations would, I proposed, make simple repeats more complex in a random way. For example, a sequence of GAGAGAGA might change into a sequence of GAGATAGATAGA if 2 point mutations occur subsequently. These longer repeating units would also be prone to slipped-strand mispairing and could therefore expand. A comprehensive survey of the relevant scientific literature, which is routinely expected for any scientist, led me to an obscure paper that had been published many years earlier. The Nobel laureate who had played a major role in the discovery of DNA polymerase, Arthur Kornberg, found that purified DNA polymerase would sometimes generate simple repetitive sequences in a template-free manner. Kornberg observed that simple sequences combined with nucleotides and DNA polymerase in a test tube readily generate long, repetitive sequences, starting with a short DNA template. In other words, Kornberg observed DNA polymerase slippage events that are similar to those that I hypothesized take place in living cells. In my case, this hypothesis resulted from my analysis of widespread simple repetitive sequences in databases of genomic DNA sequences. Synergy between replication slippage and unequal crossing over. Unequal crossing over is a process that occurs between chromosomes during meiosis when sex cells are produced. This is another mechanism of great importance for DNA sequence evolution, not only for production of repetitive sequences, but also because large segments of chromosomes, often consisting of entire genes or even many genes, are replicated in this way. Synergy between tandem repetition and gene duplication. In my doctoral thesis work, I also noted what had been suggested by others, namely that simple repetitive sequences would be prone to unequal crossing over, which plays a major role in gene duplication. Gene duplication refers to long sequences of DNA containing both protein coding regions as well as repetitive sequences. The following sections will help to illustrate the major importance of this mechanism for genomic evolution. Global significance of simple repeats. The hypothesis of simple repeat evolution by SSM combined with point mutations has stood the test of time. Simple repeats often expand at higher rates than the frequency of point mutations, which makes them an important source of variation and expansion in genomic DNA.